So, so I think you remember fusions in that we discussed about translational fusion and transcriptional fusion. So the basic, I'll just put it as fusion. So there are when you fuse two things and you want them to be expressed together. Say you have two genes, you put them up like this, and here is a common promoter. Say this is, I will just try once again and let's see how it goes. Say for example, there is gene A and here is its promoter, and you may also have gene B like this. Instead, if you are going to put them together like this, gene A and gene B in one as one operon, right? There are two genes here, gene A and gene B, and they are expressed from a common promoter. So if it is upon transcription, what you get? You'll get an mRNA, and the mRNA would have, sorry, would have the open reading frames for gene A and gene B, and upon translation, we would get two proteins, protein A and protein B. Okay, we are making, when you say transcriptional fusion, this would be transcriptional fusion. Okay. Then let's see what is translational fusion. Then you would understand. And in translational fusion, say you have gene A and gene B, and you want it to be, you wanted translational fusion. In that case, we have to do something like this. We'll have one open reading frame. Sorry, we'll make, this is assume gene A. Gene A would have, say, on DNA level ATG, and it should, open reading frame should have a stop codon, say TGA. Before that, before the stop codon, say you have AGC, there is another codon. Okay. Now you have to make a fusion. The fusion for gene B will have its own ATG and TGA, say, stop codon. So when you say in frame, this is the reading frame. It starts in multiples of 3. The first codon is 3 bases. Then you will have many of them. And then you have this penultimate, that is 3. And then the stop codon, which the ribosome will read and terminate the translation. The same will be for the other one as well, for gene B. So now you have to put it in such a way that, or yeah, let me say, the stop codon is removed and gene B has its ATG. That should start right away from here. You necessarily don't have to have gene A, sorry, the start codon for gene B, right? So here is ATG of start codon of A. You remove the stop codon, so you have something like AGC, this one. And then you're going to put, you're continuing the next one of the start codon of gene B, and you will have TGA. So this is now one complete ORF. It starts with A, and there are continuous codons of 3 bases, and eventually stops at TGA. And when a protein is produced, it produces something like, it has A and B. This is a fusion protein. This is in frame. Okay. I will just describe what is out of frame, and then you can compare with what we already made. So I am putting back another, say, ATG of gene A. And here you have AGC. Okay. And because of some mistake, assume that this A is missing. And now I'm going to have TG and whatever bases are there, and eventually happening TGA. Now, the ribosome will start reading bases in threes as codon. Until here, it read fine. And from here on, because one is missing, TGC, or say assume something else is there, it will start reading it in a different frame as opposed to what B should have been read. Did you understand that, Pratana?
Yes, sir.
That is out of frame. Okay.
Okay,
So
okay, sir.
when we are fusing the two open reading frames, one has to ensure that the reading frame is in proper order. Even if you miss one or more of bases, the reading frame is altered and it is no longer B. Something else will be produced. A part will be produced, and there will be something else that, but not B. That is, it is something like frame shift mutation.
Okay, sir.
Okay.
Y-
Where did you get this question? And give me the correct context. So probably I'll be
Sir,
able to.
I got you. I actually mentioned it in one of your videos, sir. You mentioned in frame and out of frame, so
Okay.
yes,
Yeah,
sir.
but this is what it meant.
Okay, sir.
Yeah.
Okay, sir.
I think somebody else wanted about Splinkret PCR before that, and inverse PCR. We have discussed. Do you remember inverse PCR?
Yes, sir.
Yeah, I'll just give so that hopefully this video will be useful for somebody. So, if we are doing any screening, this is the genome, and we don't know where a particular gene is there that confers a particular phenotype. We don't know what the gene is. So, what we will do is we will overexpress or try transpogenesis, or using transposons, we can create mutants. So, and then we will select the mutant that has lost the phenotype that we are looking at. That means it must have lost the phenotype because the transposon may have insertionally inactivated that particular gene. So we got the mutant during by that, and from mutant, how can we identify the gene? How can we identify the gene is the question. So for that, we discussed about inverse PCR. In that case, we should have used a transposon whose sequence is known. So we should have known the sequence of the transposon. If we know the sequence, we know what are all the restriction sites it has, and we can also design primers. There are two advantages that they give, right? So what we will do is we will then use this transposon, create the mutant, then we are supposed to identify the gene in which the insertional inactivation by the transposon has happened. So you'll isolate the genomic DNA, and then we will digest it with one of the restriction sites. So assume that in the transposon, here I think I have used GATC based ones, right? I think I described it well in the other one. The more technical details were given in one of the videos. You should be able to watch that. So we can choose a restriction site that is not present in the transposon. Say this is BAMH1. We can use EcoR1 and just giving. And then we will isolate that. So the genomic DNA will form several fragments. Some of them will have the transposon like this with the gene of interest. So all the ends are EcoR1 ends. Then we should add ligase. When we add ligase, they will circularize like this, and the one with the transposon will also circularize, something like this. Now we are supposed to treat it with something like BAMH1. In the next slide, there are figures. We will watch that. But when we use BAMH1, we would have, if there are, these will become smaller pieces. This will also become smaller, and this will get cut something like this. And in that process, what happens is if it is linearized, what you'll get is this is the transposon part, this is the gene part, and this will be the transposon part again. This was actually like this, but because of ligation and digestion, the gene parts, have become in inside, and the transposon parts are outside like this. Now, because you know the sequence of the primer transposon, we have designed primers. We can use that and go for Sanger's sequencing. That is how we can identify the region in which the insertional inactivation has happened. Here is the here is one of the slides that will illustrate the same, but in this is with Drosophila. There will be a little bit change. The overall principle will remain the same. So we have made a lot of we have made mutants, out of which we isolated the mutants, and then now we are supposed to find out which gene is inactivated. So we'll use restriction sites to cut it, and then we will get several fragments like this, and we know the sequence of the transposon. This is the transposon that is put, and then we will ligate, and then we should we can use prime PCRs and to get a amplicon, something like this, and then go for sequencing. Here is another alternative, or actually a better one. We will see that is called as Plinkeret PCR. And first thing before going, we should understand the setup. For that, we will see what is this Plinkeret here. It is basically something like a linker molecule, or a linker or adapter molecule, you can call it anyway. Only thing is it has top strand like this, and at the end of the top strand, the 3' end of this top strand can fold upon itself like this. Whereas the bottom strand is something like this one, long one. Okay. There is an interesting way of how you can see it. Of course, this is with the GATC. I mean, this must have been a GGA TCC, right? BAMH1 side. So I hope you understood this one. Why did they make this hair loop or intramolecular base pairing? Is something like this end of this adapter is not cannot be ligated to anything else, right? If you remember, we learned about linkers, is that if we have a linker like this, and if you have a DNA molecule and we add linkers, many linkers could add up like this, right? That is because on the both sides they are vulnerable to, I mean, ligase can recognize them and add them up. Instead, one of the sites you can put something like this. Now this is not a substrate for ligation, so you can ligase cannot act upon it. That is one of the reasons or intellectual design of these Plinkeret PCRs. Now look at the scheme of it. So we have had the mutant here, the same way as we were doing it with inverse PCR. Now we have a transposable element. We isolated the genomic DNA and digested it to the restriction sites. And now we are ligating the Plinkeret. Right? Plinkeret PCR, it is depicted hairpin. And now we are ligating the Plinkeret. Right? Plinkeret PCR, it is depicted with this hairpin-like thing here. So on both sides, they will add, and nothing else can be added on here or here. Nothing else can be added because those ends are not substrates. Now, when we design the PCR, the Plinkeret, we also, I think I forgot to mention that, we also have this sequence here. This is S1 primer, like here, depicted here. And this is S2 primer, depicted here. This is nested PCR. This is for nested PCR. I'm unsure if we have discussed, if you remember about nested PCR, it is a simple concept. Only thing is if you have a DNA and you have a gene here, and if you're doubtful of that it's not, the primers are not very specific, you will have primers designed like this, saying, and you will do a PCR. So you'll get a product like this. And the second set of PCR primers are somewhere inside like this. So even you will say P1 and P2. You'll use once to get this product. You will use this product as a template and then use P3 and P4 as primers. It will add specificity to it. Then when you do PCR like this, you call it as nested PCR. Okay. So for that purpose, you can use T1, T2, that is the primers that are in the transposon. Here, S1 and S2 are in the splinkeret that we are added. We have ligated. Now we can use T1 and T2 or T1 and S1 and do a PCR. And you can also use T2 and S2 and do a PCR. Additionally, you can have a final where you can use T3 with S2 and perform amplify the sequence and go for Sanger's sequence. Okay. Say the question that should be there is, if in a fragment that does not have um, the transposon, what will happen? Say here it got this blinkeret, something like this. Okay. This part of it is the splinkeret, and this is the DNA where transposon is not there. And I'm comparing a fragment with the transposon and a fragment without the transposon. What will happen? In that case, S1 is here, and you added T1, right? You will not get a product in this case because this fragment that we have is not having this transposon. No transposon, so no amplification from this side. Okay. But if you have S1 here, then you might have a product, isn't it? And in that case, if it happens, you know which one is the product. T1 and S1, if they will form a smaller product like this. S1 and S1, because you're ligating the whole splinkeret here, and it will ligate on both the sides. It will form a larger fragment. Okay. And that is one of the reasons why you use T1, T2, and eventually T3, which are all specific for the transposon to amplify and then go for sequencing to identify which region, which gene has got mutated. Okay. Any questions in this, or you want me to explain something else? Pratna, are you around?
Sorry, sir. There's some problem with my internet, sir.
Okay.
I Sir, when the splinkeret is attached, won't it hinder the process of PCR when the primer is on the other side?
And the primaries,
Won't there be a necessity to add a primer on the other side for the opposite direction PCR, sir? Because
this one.
yes, sir. So if this strand is being extended, won't the other strand also be needed to be extended?
But they will get denatured, right? What do you mean? You have
Sir,
this is it.
I'm not able to understand the step where T1 after T1 and T2 and S1 and S2 is added. I don't understand how PCR will take place.
In this case, how
Yes, sir.
PCR will happen. What is the problem here? I'm drawing here. You can see that, right? I'll say T1 and say S1. I'll put primers there. What's wrong with that? A denaturation step. This will get extended this way, and this one will get extended this way.
S- So the splinkeret will be excluded in the final sequence.
No, there is nothing called. There is nothing excluded. It can add up. Only what you have to see is first we are amplifying with S1 and T1, right? Okay.
Yes.
You have to see the design here. See, S1 is from in this part. Okay. S2 is internal to this hairpin loop. somewhere here. This loop will be gone when you use S2 and T1 T2. I mean, why is this a problem?
Oh, okay, sir. I didn't realize the loop will be gone. I just
No,
assumed it's going to stay.
even if it is there, how will it be a problem? In
S-
PCR, we are using a high temperatures. All the hydrogen bonds are supposedly gone, will be gone.
okay, sir.
Okay,
Okay, sir.
this we should. This is supposed to. Yeah, let me check once again. This sequence will actually base pair with this one, right? Should base pair with this, not with this one. If it is blink one, because 5 prime to 3 prime and 3 prime extension will happen in this way. T1 will bind to this part, this the lower strand, and will synthesize this way, and the other one will bind here and synthesize. What you need to understand is simply is if the primer is not if the in PCR we are using high temperature, hydrogen bonds are not a problem. All these bonds here or here anywhere. But
Okay, sir.
the main purpose, I think, is to avoid any kind of ligation, further ligation upon this.
Okay, sir.
Okay,
Okay, sir.
that is the problem. Yes. Any others?
No, sir.
Okay. Any other question? If not this one.
No, sir. That's all.
Okay. How is preparation so far?
S- Fine, sir.
Is today's 11th, 12th, 13. We have class tomorrow. And will that be the last one or what? I'll stop the recording then.
We have class tomorrow, sir.
Okay. Uh, I want to know, is there any project review or something?
Sir, reviews over for our department.
Okay. Why isn't anybody showing up? Any idea?
I'm not sure, sir.
Okay.
Sir, maybe because of the preparations.
Okay. Mm, that's all right. When did the review finish?
2nd Jan.
Okay. Oh, they finished long time ago.
Yes, sir.
Okay.